Hi, I'm John Kerr, one of the authors of this work recently published in Science Translational Medicine, which describes a new technique to monitor the health of a tissue in real time. Have you ever been totally broke? I mean, completely out of money. Do you remember how it felt? If you felt inactive or like you wanted to die, then you are just like a cell. You see, just like we need money, the cell needs energy to function and survive. And to produce that energy, the cell needs oxygen. When patients are sick, oxygen delivery can be compromised, and identifying when that occurs is vital to proper treatment. Until now, we have had to analyze blood that passes through a tissue to assess its health. Unfortunately, markers that we gather in this way tell us nothing about a specific organ we might be interested in. For example, here we are interested in figuring out how well the heart muscle is metabolizing oxygen after heart surgery. To really explain what we're talking about, let's take a deeper dive into what oxygen actually does in the body. The food that we eat and the air that we breathe ends up in a specific part of the cell called the mitochondrion. Specifically, energy from food is transferred to a group of proteins in the mitochondrion in the form of high-energy electrons. Conceptually, these electrons bounce down an energy staircase, a series of protein complexes called the electron transport chain. With each bounce, a proton is launched into the inner mitochondrial membrane, creating an electrical gradient that is used to generate ATP, the cell's currency. Oxygen's role in this process is to remove the electrons from complex 4, allowing the process to continue uninterrupted. The more energy the cell needs, the more oxygen it needs. And normally, the body does a fantastic job meeting this demand. But sometimes, things go wrong. Oxygen delivery may be compromised by a blocked vessel, like in a heart attack or a stroke, or it may be impaired from swelling after heart surgery, for example. In these situations, the bouncing balls build up on the stairs, wreaking havoc on the cell. Not only does the cell not have enough energy, but this state may be the trigger for cell injury and death. Now, in reality, of course, we're not talking about tennis balls and stairs, but a series of complex proteins. We used a technique called resonance Raman spectroscopy to determine the fraction of complexes with electrons on them. This creates, for the first time, a measure of how adequate oxygen delivery is relative to its demand. To do this, we shined a very precise laser at the heart. This has about the same intensity as a laser pointer. Using vibrational spectroscopy, we were then able to distinguish how many complex 4 proteins are in the reduced, meaning having an extra electron, versus oxidized state at a given point in time. In health, we found this fraction is about 20%, a number that was consistent across species. We found that when the reduced fraction increases above 40%, the cell is in trouble. Specifically, we found that we could predict which hearts would stop functioning completely, a lethal condition known as cardiac arrest, very accurately in animals by monitoring the mitochondria on the surface of the heart. In the future, this technique might be used in a variety of settings, including in the care of patients after an accident or while undergoing a major operation, or to help us better understand cancer biology. For more information, please feel free to contact us. Thanks for listening.